Bismillahirrahmanirrahim. 9700 on request of one of the students. I'm doing the June 22 paper 5-2 and this is the question 1 being solved. As you know, I've been talking about in the previous videos as well. This is a paper which needs a lot of thinking and planning. So this is for 1 hour and 15 minutes and this is on the same day as the paper 2. And in the paper 2, you know, that is also for 60 marks. This is only for 30 marks, but the time is the same. That paper 2 is also for 1 hour and 15 minutes. Now, why there's so many marks for paper 2, same time, but more marks? Because it's not a very thinking paper. In this, we have only 30 marks. This paper is only of 30 marks, but why is it for 1 hour 15 minutes? Is because you need to think a lot and you need to read the question a number of times and then plan what you're going to write and how you're going to write the answer. Now, as we start reading the first question, it's figure 1.1a. Figure 1.1a shows the root vegetable beetroot, Beta vulgaris. Figure 1.1b shows a single cell from the beetroot tissue. So this is the single cell from the beetroot tissue. The vacuole, this is the vacuole, which contains betalain, which contains the red pigment betalain. The pigment molecules are too large to diffuse out of the vacuole. Vacuole has got a membrane, which is the tonoplast, so it cannot move out of the vacuole. So this cannot come out. It cannot come out. So it stays inside the red pigment, stays inside here. And this is the red pigment, and it cannot move out. Why? Because it's too large. Look at everything in the question. Now this is the red tissue. This is one cell. This is a single cell from the beetroot. Now I want you to look at the beetroot. This is what the beetroot looks like, and I am sure you must have eaten it. I mean, we have it in our salads, we boil it, and then we can also have beetroot juice, which is very good for health. So this is what a beetroot is. In Urdu, it is called chukandar, and I'm sure you must have heard of it in whichever country you're living in. When pieces of beetroot are soaked in a solution of calcium hypochlorite, the beetroot tissue changes from red to white. This is caused by the calcium hypochlorite which diffuses into. So the calcium hypochlorite diffuses into the beetroot, diffusing into the beetroot cell vacuoles and decolorizing the betalain pigment. Figure 1.2 shows a cube of beetroot. So what they did is they took the beetroot and they took this cube, cut a cube out of it before soaking in the solution of calcium hypochlorite, and the halved cube of beetroot after soaking in the solution of calcium. So then we cut it into half. So then we've cut it like this. We've cut it from the center. Beetroot cube before soaking, beetroot cube after soaking, width of the red zone. This is all the red zone. This is all the red zone, and this is so I'm going to use a black marker, and this is the zone which has become clear. So the red zone, beetroot cube after soaking, halved after soaking, so there's the red zone, and then this is the decolorized zone. And this is the width of the cube. So we can easily measure this. How much is this measure? From here, from here, from here. We can measure it. Sometimes it could be the same, could not be the same also. You think of it. I always say to you, when you're doing the paper 5, think you're doing this in the lab. So what would you do? How would you do this experiment? Okay, as we go on, now as we read in the question, in a class experiment, cubes of beetroot of different dimensions were placed in a beaker containing a 10% solution of calcium hypochlorite. So we took, in a beaker, we took the hypochlorite, and in that we put a cube of the beetroot in it. And we must have put there's a certain volume of the hypochlorite, which you must think. Whenever there's a liquid, think of the volume, and the volume is always measured in cm cube. The cubes were left for 30 minutes. Now that's the important thing you remember when you go to read the planning, then you're going to be talking about that. The cubes were then cut in half, and the width of the tissue remaining red, the red zone. So if we had, this was the cube, and after 30 minutes, if this was the red zone, then what did we measure? We measured, we cut, and the width of the remaining red zone was measured. So totally in the beginning, this whole thing was red. And then after 30 minutes, when we took it out, then it wasn't red. This part wasn't red. So we had some part of it going away and we had this part left. So the remaining red, the red zone was measured. So we measured this then. We measured this length. We knew this length because we'd cut the cube to this size. So a group of students investigated the effect of changing the surface area to volume ratio on the diffusion of calcium hypochlorite into beetroot cubes. The student used cubes of different sizes and measured the width of the red zone after soaking the beetroot in calcium hypochlorite. Then the student suggested the hypothesis. What was the hypothesis? The width of the red tissue. This is the red tissue. The width of the red tissue remaining in the beetroot after soaking in calcium hypochlorite is inversely proportional to the surface area to volume ratio of the beetroot cubes. So we have to understand this. It says the width of the red tissue is inversely proportional. So larger the surface area, lesser the red tissue left. Larger the surface area to volume because it's saying inversely, it didn't say directly proportional, it says inversely. So if we are increasing the surface area, then this red space or this width of the red tissue is going to decrease. Larger the surface area, lesser the red tissue left inside. Now it says identify the dependent variable. Now I've talked about this many times, dependent and independent. Dependent is the result. What are you measuring? What are you doing? Not the process. What are you measuring? So what are you basically measuring is the width of the red zone. So width of the red zone, but actually you're not, you sort of, we are studying what? The surface area connection with the width of the red zone. So width of the red zone after soaking. So this was the 1 mark that you're going to get for this. Then we go on to the next part of the question. It says sketch a curve on figure 1.3 to show how you expect the width of the red tissue to change as the surface area to volume ratio changes. If the hypothesis is correct, label the axis. Now I want you to look at this. How a 1 centimeter cube, the surface area is 6:1. Then a 2 centimeter, the ratio is 3:1. So as the size increases, and then it is 3:2. So how do we calculate the area of the side, then the area of the cube surface, then the volume, and then please pause the video here and have a look at this and see how we do these calculations. So the highest was here, 6:1. Then it's 3:1. Then it's 3:2. So the smallest has the largest surface area to volume ratio. How do we calculate this? Everybody must know how to calculate the surface area and the volume. Please pause it here and have a look at it again. So very easily understandable. Now we have to remember is that the dependent variable always goes on the y-axis. So the dependent variable, which is the width of the red zone, will go onto the y-axis, and that should be in millimeter, and the surface area to volume ratio will go on the x-axis. Now you get an inverse relationship. You see, the more the width of the zone will be when there is less surface area. The surface area is increasing like this. So the larger the surface area to volume ratio here, the lesser will be the width. Larger the surface area, lesser the width. And as the surface area, sort of decrease, this is now as it larger the surface area to volume ratio, this is larger the surface area to volume ratio, the lesser will be the width. The width is increasing here in this way. So we can just add some figures in it, 1, 2, 3, 4, 5 or something like this. So you do just write some numbers on it and then you can easily figure it out. Then it says B1, describe a method the students could use to calculate the data needed to test their hypothesis. The students were provided with beetroot tissue, a 10% calcium hypochlorite solution, and standard laboratory equipment. Your method should be set out in a logical order and be detailed enough for let another person follow it. Now you can see this is for a lot of marks. This is for 7 marks. So this is usually 7 or 8 marks, and this is what we need to think about it. Now, as I say very often is, and I say it in school as well, is that think you're doing this in the lab. So you have the beetroot and you have the 10% calcium hypochlorite. Now what are you going to do? Number one, first, you use the same beetroot. You can't be changing the beetroot. So you use the same beetroot. You take 5 cubes with different sizes. Okay, what do you mean different sizes? You have to cut cubes. So you can take them of maybe 0.5 centimeter or 1 centimeter to, so the range which was you were supposed to take, because you remember beetroot is not such a big vegetable. It's a very small vegetable. It's just size of a potato or even a small potato. So you could take something about 1 centimeter or 2 centimeters. So 0.5, 1, 1.5, 2, 2.5 and something like this. You could have given me these figures. Any figures you could have given me. Then you would use a scalpel and a ruler to cut and measure, to measure and cut actually. Then you would calculate the surface area to volume ratio. Then you would ensure that you would put enough calcium hypochlorite to have them fully covered, fully immersed in it. Then you would cover the containers because you don't want them drying out after soaking. Then you cut in half with the scalpel and you measure the width of the red tissue with the ruler after soaking it. Then you repeat the experiment minimum of twice and you calculate a mean. You must say calculate a mean. Please do not use the word average. That's not allowed. So at least 2 readings and calculate a mean. And then of course you can talk about the hazard. Naturally hypochlorite is a chemical, so it's harmful. You use gloves or the scalpel. You have to turn it away from you. The sharp side should be away from you so that you don't cut your finger or you cut on a tile. And the beetroot, you could be allergic to beetroot, and you just need to wear gloves when you're handling the beetroot. Now, as you can see, I have now written this down, and you can also write it in the point form. It doesn't say can't write it in the point form. Please remember, you can write in the point form. You can think in the point form. Use same beetroot, then take 5 cubes of different sizes, and then you might give me the sizes 0.5, 1 centimeter, 1.5 to 2 and 2.5 centimeter. Then use a scalpel and a ruler to measure and cut. Remember, you have to talk about the equipment, the laboratory equipment. So use a scalpel and ruler to measure and cut. Then you calculate the surface area to volume ratio. Then you keep the cubes fully submerged in the solution. Then you remove after 30 minutes. And then you maintain the temperature using a water bath after the cubes are immersed. Then you use a tissue to dry the cubes after soaking. Then you cut in half and measure the width of the red tissue using a ruler. You must tell me how you're going to measure it. And then you repeat twice, at least twice, but you should say, I always say write more, repeat 5 times and calculate a mean. And then you have to give me, the important thing is there are 3 things that you have to think of: the hazard, the risk, and the precaution. So the hazard is calcium hypochlorite. It's an irritant, or you can say it's harmful, or you can say it's toxic, or you can say it is corrosive. So that's the risk. What is the precaution? You use gloves. You could have also talked about the scalpel, so it can cut your skin or can injure your skin. That's the risk. And so you cut it away, or you cut it on a tile, and the scalpel should be facing away from you. Then the beetroot, it's an allergen. You could be allergic to beetroot, and so that is why you should maybe you can then use gloves while handling the beetroot. So any plant matter, you can say this, it can cause allergies, it's an allergen, and you use gloves when you're handling it. And I always tell you this, and I'm allergic to mangoes, and even if I touch the mangoes, I can get a rash on my hands. So I know it's a weird thing, but that does happen. So hazard, risk, and precaution. So here, of course, I've said the hazard is calcium hypochlorite, and the risk is it's an irritant, and the precaution is use gloves. So I'm not saying you have to give me all these points. There are 12 points, and you can have to give me any 7 of them to get your 7 out of 7. Then coming to part 2 of the question, the students decided to calculate the percentage change in volume of the red tissue in each cube. State how the students could calculate the percentage change in volume of the red tissue in each cube. Now that's easy. Initial volume minus the final volume divided by the initial volume and into 100. So initial volume minus the final volume. So we have the initial volume minus the final volume. We are saying percentage difference. This is a very, you have studied this, I think, in maths or something, divided by the initial volume. And then you multiply this by 100. So this gets you your 2 marks. The 2 marks for this. 1 mark is for this formula if you wrote that, and the other mark is for multiplying it by 100. So initial volume minus the final volume of the red cube and divided by the initial volume of the red cube and into 100. In a further experiment, the students investigated the effect of temperature on the release of betalain pigments from the beetroot tissue. Cubes of beetroot were cut, beakers containing water were prepared. The beaker were placed in water bath at temperature between 0 and 85 degrees Celsius. The beakers were allowed to equilibrate for 5 minutes. They need to come into equilibrium with the environment. The beetroot cubes were added to the beakers. After 30 minutes, the beetroot cubes were removed. Colorimeters, each with blue-green filter, were used to measure the percentage transmission of light through the fluid remaining in each beaker. So what are we studying? The effect of temperature on the release of the betalain pigment. So more the release, more darker the red pigment, and the colorimeter is going to register that. The results of the experiments are shown in table 1.1. The student decided that two of their results are anomalous. One of the results that they consider anomalous is circled in table 1.1. Is this one here. Circle the other anomalous results and complete table 1.1 by calculating the mean percentage transmission of light at 25 degrees Celsius. This is the one which was not done, so you're supposed to calculate this. Now, which is the other anomalous reading is the, when you look at it, it doesn't tally, is this one, 17 at student 4 at 65%. So this is the one which doesn't, you see, you've got 26, 31, 29, and then a 17. So there was only one which was an anomalous reading, and then of course the calculation here was 89.3. So 89.3, you did the calculation of adding all of them up and then dividing it by 4, and this was 89.3. So this was a very, very easy 2 marks for you to get. Then coming to part 2 of the question, describe 2 ways in which the method described in C could have been improved to give better repeatability between the results of the different students. So method in C which you talked about was the temperature thingy. So all cubes of the same size, or you can say all of the same surface area to volume ratio, then the same age of the beetroot, sufficient water to cover the cubes completely, then same volume of water in each beaker, then same colorimeter, then you stir before taking the sample for the colorimeter reading so that the red color doesn't settle down. Then check the temperature of water in the beakers have equilibrated. Check your temperature of water in the beakers have equilibrated. Then zero colorimeter calibrate before each reading, and use a thermostatically controlled water bath. So out of, you only have to give me 2, but I have about 9 points which you could have easily given me any 2 out of these. So be very careful when you are writing these so that you get your 2 marks out of this. Same size, same age of beetroot, same volume of water in each beaker, same colorimeter, zero colorimeter before each reading. You calibrate it. You take it to 0 every time, and then you use a thermostatically controlled water bath. Then coming to the last part of this question, with reference to the data in table 1.1, state and explain the effect that increasing temperature has on the release of betalain from the beetroot tissue. So let's look back on this. Now, when it was 5 degrees, the percentage transmission of light was 97.8. And as we have increased the temperature to 85, there's only 1.3 is the percentage transmission of light. Why? Because it's becoming a darker red. As the temperature is increased, the proteins in the phospholipid bilayer are denatured, and all the betalain is going to diffuse out. The cell membrane is going to lose its shape or its structure, and this can be, these large molecules can move out. So 85 shown you 1.3, so there's very little transmission of light. That means it's very, very dark. As I always say, water allows all the light to pass, but if you put Pepsi or Coke in it, it's not going to allow light to pass. So the darker the color, less light will be passed out. Only most of it will be absorbed. So percentage transmission will become less. Most of it will be absorbed. Colorimeter tells you the transmission of light, how much it is absorbed or how much can pass through it. So now if you look at this, so then how are you going to interpret this? It says more betalain released as temperature increased. So, or you can say as temperature increases, more betalain released. Why? Because the membrane fluidity increases as temperature increases. Or you can say the membrane proteins denature at high temperatures. So as temperature increases, more betalain released, membrane permeability increases as temperature increases, and membrane proteins denature at high temperatures. So this was a total 18 marks, and the next question is for 12 marks. We will continue this in the next video, and please do let me know how and what is the things which are you not being able to do in these paper fives. These are very difficult papers, and you have to make a great effort to look at the study the paper, then look at the exam report, then look at the mark scheme, and then understand what was expected of you and how do you word your answers. Thank you very much.